one of the topic is directly related to industry, how we can utilize the functional annotation and into animal breeding applications. So Derek Bickhart from Hendrix Genetics going to give a talk about how integrate of the functional annotation into the animal breeding. Derek, yours.
Thank you very much for the introduction. I may have an unstable internet connection. Hopefully that is not the case. Please let me know if you lose any part of my presentation. But I'd like to begin by discussing the topic. And as mentioned, the goal here is to talk about how industry is currently using the information that is provided by these large consortia that are looking into functional annotation. It is a very useful resource. And I'd like to demonstrate this with two applications that me and my team have actually looked at. The first is a more classical causal variant screen using functional annotation data to identify causal variants that are causing disease or production phenotypes in animals. The second is to incorporate functional annotation data into quantitative genetics models to try to breed for better performance from animals. And finally, I'd like to tie it all in together to talk about how we can better integrate both industry and academia in future consortia to generate this data, and also new innovations that are coming down the pipeline that will improve the collection of this data in the future. So first, I'd like to start with an example of some of the classical techniques that me and my group have published several times or have incorporated into our breeding programs. And the real problem that we're trying to address here is the fact that many of the variants that affect our populations tend to be low frequency but high effect genetic variants. These are often easy to miss given our reliance on SNP genotypes that often do not track low frequency variants in populations. And also because of their low frequency, they tend to be more difficult to impute using selected reference panels of individuals. They're often left on the side, but could have deleterious effects on our populations. And given their low frequency, we want to keep it that way and to identify them sooner before they become a major problem within our breeding lines. The Human Decode project gives us some idea for how frequent these could be within our populations. Within human, the Decode project identified over 500 loss of function SNPs and indels that had minor allele frequencies lower than 2%. Many of these affect genes that are involved in expression within organs and other tissues. Within animal populations, though, there's an important difference. In some species, we use patrilineal breeding programs, which can actually exacerbate the problem and help propagate these variants more widely. And to give you an example from my previous career working in cattle, there was a highly influential Jersey bull called Observer Chocolate Soldier, who was used in the 60s and 70s extensively in breeding programs for the Jersey cattle breed. Unfortunately, Observer Chocolate Soldier was a carrier for two recessive lethal alleles, in which case, if you are a heterozygous carrier, there's no phenotype, but homozygotes die as embryos. Because of the production traits that Observer Chocolate Soldier gave to his progeny, he was used quite frequently, or his resultant children, and this ended up increasing the frequency for those recessive alleles up to about 20% in the Jersey population at one time. This resulted in devastating losses for the breed, with the frequency of fertility dropping by 40% for carriers in that breeding program. It wasn't until 2012, 50 years later, that we identified the causal variant that was actually responsible for this allele. And it turns out it was a single nucleotide polymorphism, or SNP, that changed a cytosine base to a thymine within the coding region of a gene. That gene was CWC15, which is an important gene in the spliceosome, which results in the splicing of messenger RNA for protein translation. This is essential for development. And you can see from the protein structure analysis on the right the exact cause of this mutation, it truncates the entire protein almost in more than half. We need a system to predict these more frequently. When we're dealing with coding sequence, it was quite easy to identify this loss of function as a major effect. The reason for this is that we can predict, based on known rules in coding sequence, how a mutation will affect the translation of a protein product from a gene. And you can see on the left, the gene sequence is broken up into the codon bases, and each codon translates into an amino acid. In the top and the bottom panel, the sequence is the same except for a single SNP, which changes the isoleucine into a leucine in the carriers. This is more easy to predict than the opposite, which would be any changes to regulatory apparatus within cells. These changes may change the expression profile of a gene in ways that are not immediately easy to interpret, either through increasing the gene transcription or decreasing it, which may result in deleterious effects. Here, important work has been done on the functional annotation aspect to identify expression QTL, or quantitative trait loci of gene expression, and requires extensive functional annotation data sets and lab validation. This is why this information is useful to us. We can now identify eQTLs that may have an effect on gene expression, and if we notice this frequency increasing in our population, we can do further experiments to see how it's affecting our product lines in industry. I'll spend a lot more time discussing the ways that we've tried to incorporate functional annotation into our breeding program. And here I must give a lot of credit to my colleague, Bruno Perez, also of Hendrix Genetics. Bruno implemented a new system that was developed by Mike Goddard's group called FAITH scores. FAITH stands for Functional and Evolutionary Trait Heritability. But in short, it is a way that you can identify SNPs or other genetic variants, and then estimate the amount of genetic variants that each of those variants explains for phenotypes and functional annotation. And I won't have time to talk about the exact methodology, but please remember that this is a consolidation of all of that information to run a breeding program. So there is a bit of an averaging in order to make this work in practice. In order to test this and to produce this, we sequenced, as part of the Gene Switch Consortia, 11 landrace pigs that had extensive whole genome shotgun data as our reference panel. We then used that reference panel to impute 25,000 SNP genotypes that we had on other individuals, and ended up with a total set of about 15 million SNPs that were imputed across the entire population. For our phenotypes, we used 6 traits that were recorded by Hendrix Genetics and other partners on the landrace breed. And we had about 6,000 to 22,000 records per trait across the entire population. In order to add the functional annotation layer that's required for the FAITH scores, we used not only publicly available data that was accessible to the consortia, but also data that was generated as part of the Gene Switch and the Pig GTEx consortia. In total, we used 32 functional annotation data sets in order to generate the scores for each variant type. This is the list of functional annotation layers from the consortia themselves. And this is a very big slide, so you're not expected to read all this. But I would like to note that the top 4 data sets were directly generated by the Gene Switch Consortia and their partners, and the bottom were produced by the Pig GTEx Consortia. From public data sets, we use the following information, including previous annotations of things such as zinc finger binding regions, active transcription start sites, and enhancer loci. So we had a large data set of functional annotation that was both publicly available and generated by ongoing consortia to use. We'll now move into some of the results of the testing and the FAITH score analysis. And the first and obvious benefit of FAITH scores is that they can estimate on a per variant basis how much of the genetic variance is explained by that variant. And here on the right, you can see a range of per SNP heritability across all of the traits for each of the different functional annotation layers. To break it down even further, it turns out that many of the eQTL loci, including the BIQTL, which stands for breed-specific QTL, ranked very highly in terms of capturing the proportion of variance. I would like you to also interpret the range of the variance as indicating that there's much more trait specificity in terms of the proportion of the variance that's captured here. So if the variance is very high, some traits actually had lower performance than others in terms of the heritability. Also, what was really great is that this kind of confirms some of our molecular hypotheses as well. You can see the expression QTL located at the top in green, and then some of the more transient epigenetic markers, which are not necessarily inherited through the same way, being accounting for far fewer of the proportion of the variants down at the bottom. These are indicated in purple. Also, some of these conserved sites that have lower standard deviations are more likely to be trait independent, so they're going to be more reliable across all of the phenotypic traits that we measure. This is another way of looking at the same data that was present on the other slide. And here is just to show you the exact proportion of the variance explained by each trait. It's a little bit more difficult to see, but in the third row, there's the breed-specific QTL listed. And you can see that for trait 3 and trait 1, it's a very high proportion that's explained. And I would like to note that as we add more functional annotation data sets and more phenotypes to this list, it's more likely that these deviations might increase and more proportion of the variance might be captured. So there's always room for expansion here to improve the quality of the faith scores that we're generating. Next, I wanted to talk about using faith scores to identify better genetic markers. As many of us know, when we sequence an individual and call genetic variants, we're often left with a very large list of potential variants. This is impractical for us to use in genomic selection. So we wanted to see if faith scores could be used to prioritize them as future genetic markers. And Bruno conducted a comparison between markers that had high faith scores versus low faith scores, and then also collected a random sampling of variants distributed across the genome to compare against the high variants as well. The questions answered here are, is the faith score able to correctly identify more informative markers than just a random chance? And also, will the high and low faith score variants indicate the proportion of the variance that is captured? These are the results of the heritability. And please note that this is conducted using the top and bottom 1 million SNPs. And for comparison, we randomly selected 1 million markers that were evenly distributed across the genome. One thing to note is that in all cases across all 6 traits, low faith scores always captured far less of the genetic variance, which is good. It means that these markers are less likely to be influential on the trait in question, and that the faith score is actually working to select more informative markers on a molecular basis. However, we noted that if you randomly select markers across the genome, the performance is the same as if you select the high faith score markers at this level. So this means that you do not get as much benefit or any benefit by selecting high faith score markers individually. We next wanted to see how this affects predictive accuracy when we try to predictively model traits based on the genotype of the individual. And again, at the 1 million SNP level, we calculated the predictive accuracy on the graph in the y-axis and for each trait along the x. Please note that the low faith score markers are always represented in red, the random are in gray, and the high faith score markers are in blue. In all cases, the low faith score markers had the lowest predictive accuracies, and this was also reflected in the previous chart. The high faith score markers, though, did show a slight benefit in certain traits, increasing the predictive accuracy up to about 4% in some cases. And more importantly, the high faith score markers never deteriorated the predictive accuracies of our selections. And here are the markers, here are the traits that did not see any increase in predictive accuracy. And these were the traits that benefited from using high faith score markers. So again, it's going to be more trait specific in terms of the improvement that's gained by using these models. Bruno also wanted to test to see if faith scores could be used in another way. So if we actually calculate the faith scores, can we use them in weighting our model? Because some markers may actually predict more of the heritability than others. So Bruno calculated values to apply to each marker individually and tried to weight it so that if an individual had one marker over another, that would reflect more in terms of the performance of the trait. And along the bottom, this is just to show the distributions of the faith scores on the left, and on the right is the weighting values that he calculated. Here, we actually tried to test this out again against the random values in gray, and also against a faith score prediction that did not have any weights. So all of the high faith score markers had the same weighting. Bruno tested two possible ways to incorporate weights in this analysis, and the first is with linear weights, which is represented in yellow, and in quadratic weights in orange, with the quadratic rates kind of overemphasizing high and low effects in the weighting. As you can see from the histogram, though, my apologies. As you can see from the histogram, there were very little benefits to using the weights in this way. And in fact, in some cases, it actually decreased the predictive accuracy on the traits. So unfortunately, it does not look like these scores can be used default in the way that we intended for weighting. So in conclusion for this section, we were very pleased with the quality of data sets that were produced by our consortia partners, and also the quality of the publicly available data sets. And both can be used to predict the values, the faith scores for these variants. We did identify an improvement in prediction accuracy using faith scores, and that the molecular QTL maps seem to be the most informative. So these, again, are the eQTLs and non-coding QTLs that impact gene expression. Unfortunately, it looks like we still need to do some additional work to see if faith scores can be used for weighting SNPs. Right now, the results suggest that they do not increase predictive accuracy. And there may be some limitations here, particularly from the use of imputation that may be driving this outcome. Now, I'd like to take a step back and talk about some future directions I see the research taking, as well as to kind of encourage the continued collaboration between academia and industry. And I'd like to be perhaps a first evangelist to saying that industry participation with these academia consortiums is a great thing, and I'd like to see more of it. And for the following reasons, I see benefits to both sides. For private industry, we benefit quite a bit more when the resources that are generated as part of these consortia are applicable to our breeding lines. To give you an example, some of the chicken reference genomes that have been produced are on broiler lines, whereas in Hendrix Genetics, we have a laying hen line, which has many different genomic features. So having relevant data sets really improves the state of prediction in private industry. But I also recognize that consortia benefit more from increased amounts of data, including those of animal genotypes. You get an improved statistical power to detect low-frequency events by having these larger data sets. So to bring together the synthesis between the two groups, I think that we need to start talking more about good frameworks in order to minimize the risk of sharing data. This is the major concern of private industry, and where we stand to lose a lot of value by losing competitive advantage with our competing companies. If we can minimize this risk, I see more potential for us to participate and to provide the data that's needed to drive innovations. I'd also like to say that some new resources are coming down the pipeline. I've done some work on the chicken pangenome to try to test it to see how much of an improvement there is. And here I wanted to test one specific metric that's important for whole genome shotgun variant calling, and that is the read mapping rate against a reference genome. And on the x-axis, you can see the mapping rate of the reads against the linear genome, which is in yellow. That's the current chicken reference genome assembly, and a new pangenome graph that was produced by Washington University at St. Louis in green. And the proportion of the variance is based on 90 chicken data sets that I aligned against both references and compared. You can see at the top that the improvement of read mapping rates is far greater for chicken lines that were from Indochina, including from the Merc T breed from India. In fact, the highest individual had an 8% increase in read mapping rate, which is an astounding amount of reads that would otherwise be missing. Whereas some domestic chickens from Bangladesh are still underrepresented in both the linear reference genome and the pangenome. In general, just using this resource without further extension, we saw a mapping rate of about 1.3% improvement, which is still a very high proportion of the genome that would otherwise be missing. I also see the improvement of generation of other functional annotation resources that the industry can use. And some of these include things such as CAD scores, which help us predict the deleterious effects of mutations on the individual. These are very important for us to use within the industry. Additionally, we're hoping to incorporate even better models in order to identify SNPs and indels that may cause production changes in our individuals, much like the faith scores. But I would like to note that faith scores currently are being used within the research community and have a benefit in other applications as well. For instance, this was a study published in Genome Biology on the comparison of differentially expressed orthologs in cattle and human. For orthologs that had the same expressed patterns, which are conserved, and those that diverge greatly, you can see that higher faith scores are found within the conserved genes. That indicates that those genes are more conserved and are more likely to be core functions. So if you find a marker that deviates from that within those genes, it's more likely to cause either a deleterious effect or an important difference in expression profile. And I also want to raise a warning to the community, and that is that we have a major blind spot. In all of the previous work that I mentioned, we were looking at single nucleotide polymorphisms, but there are many other genetic variants that are present within our populations. These include small insertions and deletions, which we've been prevented from really understanding in depth because of false discovery issues. Pangenome resources seem to improve this greatly. Another fact is that structural variation is quite prevalent in our breeds, but how do we resolve it with respect to the functional annotation maps that we're generating? If we have a duplication that covers two identified functional annotation layers, how do we predict or model the changes to gene expression that duplication may cause? These are still open questions and may require a hierarchical framework for us to better model. And finally, another open area is the concept of aneuploidy. And these can be either whole chromosome aneuploidy or segmental copies of the chromosomes. How do these impact the QTL that we've discovered? And also, what is the dosage sensitivity for certain genes at certain developmental points? More data is needed here to provide better answers to the community. And this is especially important to us in aquaculture, where aneuploidy is actually desired for certain production traits. In conclusion, I'm very, very enthusiastic about the generation of functional annotation data, and I'd like to see more industry participation here. It not only feeds forward into the detection of causal variants that can head off increases in disease or production losses in our lines, but it also gives us context for future improvements in our selective breeding programs. In addition, having more data input means that we can model traits much better using things such as faith scores or other ways to incorporate this data in our breeding programs. And as I mentioned, I'd like to see more cooperation. So I thoroughly encourage the formation of more consortia and the advanced discussions of how we can address issues from both sides in the agreements. And with that, I'd first like to give a generous thank you to Elisabetta for inviting me to present my research, and Temitope. I'd also like to thank partners at Wageningen University and GeneSwitch, and also first and foremost, my colleagues at Hendrix Genetics that did the work. And I'd be happy to answer any questions.
Okay, I start with a very nice presentation. I guess probably from their breeding company, you know, some of the information you cannot reveal, but you talk about they have 6 different traits, right? Certain of the traits seem like different. I'm just wondering for the functional annotation, did you see these biologically relevant of those tissues provide a more important functional information to improve the heritability? So for example, probably the body weight or average DNA gain might be more related to the liver annotation or the muscle annotation or adipose. Is that the case?
Yes, in fact, you can imagine that it's a lot of data to sort through, but for some of the markers that actually captured a large proportion of the variants, we took a look at the underlying molecular pathways, and it did make sense. It's not the most obvious molecular pathway for the trait, but it has ancillary effects that do influence it. So like with any other candidate gene approach, often you have a good target that you think is responsible, but really it's kind of this other networked gene that causes the effect. So it was really beneficial to take a look at these variants and see that effect. I can't go into further details, but yes, you're absolutely right. We did identify those.
Okay. The second question, I'm just wondering for the faith score, seems like you didn't see much of the advantage to compare this one higher versus random one, right? Did you see, because the faith score, what kind of the functional information you put into that index, right? And if you have more of the functional information available and added to that, did you will see that accuracy of that faith score might be have more advantage compared to the random?
Yes, and I would like to point out that this will still be trait dependent, as you saw in the previous graphs. For instance, trait number 4 had a larger predictive accuracy. But you're absolutely correct. If we include more expression QTL annotation layers, we would expect to get increased predictive accuracy there. So gaining more of these data sets on more tissue types would allow us to better refine the signal and make sure that we're capturing all the variants.
Any questions? Okay. Oh,
I think I see
one
some
question
in the chat.
Oh, Erica, you can see the question from online.
Yes. So the question that I see is, which marker is stronger for breeding selection and reproduction traits, indels or SNPs? And in what context might one be more useful than the other? It's very difficult to say. I hesitate to give a definitive answer here, especially since our picture of indels in our breeding lines is still incomplete. I think that we need to generate more better maps of indels in our breeding populations before we can start tackling this. In general, though, deletions tend to be more disruptive than single marker changes, not in all cases.
Okay, great. Thanks very much, Derek.
Thank you very much.